Thank you so much for the very nice introduction, Julia. I'm really excited to be here and present my PhD work. As Julia mentioned, I'm Shreeya, and I'm a PhD student in Professor Mika Simon's lab in Munich. Roughly 7 million people in Europe alone live with Alzheimer's disease, and these numbers are expected to rise. Typically, patients with Alzheimer's disease have amyloid plaques and neurofibrillary tangles. There's a lot of neurodegeneration as well as a lot of neuroinflammation. One of the biggest risk factors for the disease is aging. If we look at the Alzheimer's disease research field over the past few decades, it has shifted from a very neuron-centric to a more glia-centric field over the past few decades, but still majority of the research has focused on astrocytes and microglia. What oligodendrocytes do and how they respond in the disease is not well known and relatively poorly understood. And so this is what we wanted to bridge with my PhD project. As most of you might already know, oligodendrocytes are really important cells in the brain, and they perform multiple important functions, first and foremost being myelination, where they myelinate the axons, which helps increase the conduction velocity of electrical impulses. They additionally provide axons with metabolic support, giving them lactate and pyruvate as and when they need them. Additionally, they perform defense functions for the axons by providing them antioxidant protection against reactive oxygen species and iron. How do these functions change in the disease, and how oligodendrocytes respond in the disease is something that we wanted to address. To do so, we used the 5xFAD mouse model, and as the name suggests, 5 human mutations were modeled in the mouse, 3 of which were in the APP gene, and 2 of them were in the PS1 gene. One of the advantages of using this model is that things are relatively fast, which is great as a PhD student. And we have plaque deposition starting as early as 2 months. And by the time these mice are 5 months old, they already start exhibiting behavior deficits. For my analysis, I usually focus at 2 time points, one being the early time point, which is between 4 and 6 months. The second is the late time point, which is between 10 and 12 months. I started by looking at myelin damage in these mice, and to do so, we use scanning electron microscopy. I performed this at multiple different time points in wild-type and 5xFAD animals, and we looked at all of these kinds of myelin abnormalities shown here. I quantify myelin outfolds, where a small piece of myelin is bulging out, myelin infolds, where myelin bulges in, axonal swellings, where we have really swollen axons with lots of mitochondria and lysosomes, but it's still myelinated. Split myelin, where the same myelin sheet is split into 2. Redundant myelin, where the axon seems to be myelinated multiple times. And myelin fragments, where there are small chunks of myelin within the axon. I quantified myelin abnormalities in wild-type and 5xFAD animals at 4 months and 10 months, and I noticed that there was an increase in myelin abnormalities in 5xFAD animals at 10 months. The myelin abnormalities is shown in green in this picture. Next, we wanted to look at oligodendrocytes themselves and how they change in the disease. So in the brain, we have oligodendrocyte precursor cells, and they differentiate into myelinating oligodendrocytes, which further differentiate into mature oligodendrocytes. I started by looking at all the 3 cells, first by OPCs, which express the marker PDGFR alpha, which is shown in green in the panel. And we observed an increase in the number of oligodendrocyte precursor cells in 5xFAD animals at 10 months. Next, I stained for myelinating oligodendrocytes, which express the marker BCAS1, again shown in green in this panel. And we also observed an increase in the number of BCAS1 positive myelinating oligodendrocytes in AD in 10 months. Next, I wanted to look at mature oligodendrocytes, which express the marker CC1. And this is shown in green in this panel, and we observed an increase in the number of mature oligodendrocytes in 5xFAD animals at 10 months. Overall, we saw that all the 3 subpopulations were higher in 5xFAD mice. This was really, really surprising for us because we were expecting the complete opposite since there's so much neuroinflammation in the brain at this point. Additionally, we also saw something really interesting with the myelinating oligodendrocytes, which express PCAS1. We noticed that especially around the plaques, here they are in red in the panel, we noticed these weird swellings. They had abnormal morphology, they were swollen, and we had these holes there, holes in the cell, which lacked a DAPI nucleus. I quantified these swellings around the plaques in wild type and 5XFAD animals at 4 and 10 months. And we noticed that they were usually significantly higher in 5XFAD animals at 10 months. These swellings were absent in wild type mice. So in a healthy brain, we know that there are OPCs, which differentiate into pre-myelinating oligodendrocytes, which then myelinate. In ED, we have OPCs, which differentiate into these abnormal pre-myelinating cells with swellings. They are very confused. They are trying to myelinate, but there's lots of neuroinflammation in the environment. So they frustration myelinate the wrong things, leading to increase in myelin abnormalities. This results in oligodendrocyte death, which we know from literature is a very strong cue for increased oligodendrocyte proliferation, explaining why we see higher numbers of all the cells in the oligodendrocyte lineage. People, us, and as well as many different groups have reported oligodendrocyte heterogeneity in healthy as well as diseased models, including presence of disease-associated oligodendrocytes. These cells are very similar to disease-associated microglia, and they come up in all multiple different disease pathologies. They upregulate genes such as SERPIN3N and C4b, as well as upregulate genes in the immune signaling pathway. I wanted to see if this population exists in our model. So I stained for SERPIN3N in wild type and 5XFAD animals at two different time points, 4 and 10 months. SERPIN is shown in gray in this panel and CC1 in green, and we noticed an increase in SERPIN-positive disease-associated oligodendrocytes in 5XFAD animals at 10 months. The numbers were really, really low and barely even present in wild-type animals. What we also know from literature in the context of demyelinating diseases, as well as in the context of MS, that CD8 positive T cells can infiltrate the brain, and then they are myelin toxic. They can induce myelin damage. What other groups have also shown is that CD8 positive T cells are known to infiltrate the brain parenchyma of AD patients. We wanted to see if this population is this population the one that's actually causing oligodendrocyte and myelin damage in our model. To do so, we first wanted to see if there are even CD8 positive T cells in our model. To do so, we use the marker CD8, which is shown in green in this panel, and plaques are marked by thiazine red in red. And we observed an increase in the number of CD8 positive T cells in both the gray and the white matter in 5XFAD animals at 10 months. To address if this population is actually myelin damaging, we decided to deplete them by administering 5XFAD animals anti-CD8 antibodies intraperitoneally for 6 weeks, starting 6 months. This helped us get rid of all the T cells from the mice. I then looked at disease-associated oligodendrocytes by staining for SERPIN3N and CC1, which is a marker for mature oligodendrocytes, and observed a decrease in disease-associated oligodendrocytes after anti-CD8 antibodies. Additionally, we wanted to look at myelin abnormalities in these animals, so we again performed scanning electron microscopy and noticed that the myelin abnormalities were much lower after T cell depletion. This shows that T cell depletion was able to successfully rescue some of the oligodendrocyte and myelin pathology that we observed in this model. To mechanistically understand what exactly is happening and how can T cells cause oligodendrocyte and myelin damage, we decided to perform single-cell RNA sequencing for control and CD8 positive T cell depleted animals. We looked at microglia because literature has shown that T cells can really have an impact on microglia and can really activate them. What we observed was that these two microglia clusters, DAM3 and DAM4, or disease-associated microglia 3 and 4, were much lower in anti-CD8 mice. These clusters are known to express genes such as H2AA, CD74, all of which are MHC2 markers. We next wanted to see what exactly is this population, what is it doing, and can it induce myelin damage? To answer this, I first started by staining for MHC2 microglia in wild-type and 5XFED animals at two different time points, 4 and 10 months. MHC2 is marked in green. In this panel, microglia is marked in gray and plaques are stained in red. I noticed an increase in the number of MHC2 positive microglia in 5XFED animals at 10 months. This population barely exists in wild-type animals. We wanted to spatially understand what is the relationship of this population with CD8 positive T cells. So we decided to perform spatial transcriptomics. And as most of you might know, spatial transcriptomics is a really useful tool because it just not gives us the gene expression profiles of different cells, but can also give us spatial information as to where these cells are in the brain. We did this in collaboration with the Gyoshi group, particularly Peter. I looked at CD8 positive T cells, which are marked in pink, here marked by red arrows, and I looked at the microglia next to it. These microglia next to it expressed really highly expressed CD74 and H2AB1, all of which are MHC2 markers. So we noticed that CD8 positive T cells are present right next to MHC2 microglia. We wanted to see if this spatial relationship, if this relationship exists in human AD patients. So we took some postmortem human AD tissue, and I stained for CD8 and MHC2. This was done in collaboration with the Christina Stedelman group in Göttingen. CD8 is stained in brown, MHC2 in blue. And we noticed that majority of the CD8 positive T cells had MHC2 microglia next to it. This number was really high, and it was as high as 40%, which is very interesting because this number is much higher than what we see in mice, showing that these cells might actually play an even more important role than what we previously thought. But is this population that's actually damaging myelin? To do so, we decided to shift to a more ex vivo model, because in an in vivo model, there's too many things happening at the same time, and it's really complicated to delineate what exactly is causing myelin damage. To address this, we decided to first see if we can get MHC2 expression in cell culture. We know from literature that interferon gamma can induce MHC2 expression. So we took some primary microglia and treated them with interferon gamma. After interferon gamma treatment, there was an upregulation of MHC2 expression in these cells. This was further rescued after baricitinib, which is a JAK-STAT inhibitor and acts downstream of interferon gamma. Additionally, we also injected interferon gamma in the brains of wild-type animals. This resulted in upregulation of MHC2 expression in these microglia. Now, once we have a protocol to establish this expression, we decided to move to our ex vivo model to study myelin damage. How we do this is we take wild-type brains we cryosection wild-type brains, and we seed them with wild-type and 5XFAD microglia. We can then treat them with different things, including interferon gamma, and look at myelin damage by quantifying internalized myelin. Once these cells are activated, they start to eat the myelin of the wild-type cryosections and start phagocytosing the myelin. And by quantifying internalized myelin basic protein, I can say how much myelin damage there was. The more internalized myelin basic protein, the more the myelin damage. I started by looking at wild-type microglia and then treated them with interferon gamma. After interferon gamma treatment, we noticed that there were more internalized MBP in microglia. This was partially rescued by baricitinib treatment. Interestingly, when we looked at 5XFAD microglia, We could not activate this further by interferon gamma treatment because this microglia was really activated in the first place. However, after treatment with baricitinib, we were able to rescue myelin phagocytosis. This shows, so all this data pointed out that MHC2 positive microglia have myelin damaging properties. To briefly summarize what I have presented so far, in a healthy brain, we have oligodendrocyte precursor cells, which differentiate into pre-myelinating oligodendrocytes, which then myelinate. In AD, these oligodendrocyte precursor cells differentiate into these abnormal pre-myelinating oligodendrocytes with swellings, which frustration myelinate, and this results in increase in myelin abnormalities, which further leads to neuroinflammation. We have CD8 positive T cells in the brain, which can secrete cytokines such as interferon gamma, which is known to activate microglia to express MHC2. We show that this microglia population has myelin damaging properties. This leads to even more myelin abnormalities, driving a feed-forward cascade where these myelin abnormalities cause more neuroinflammation. This cascade was partially rescued by anti-CD8 antibodies and CD8 positive T cell depletion. All the data that I presented was published earlier this year in Nature Neuroscience, and you can scan this link to access the paper. To end, I would like to thank everybody involved in this project, especially Mika, for allowing me to work on the project, and without whose supervision this wouldn't have been possible, and everybody in the EM facility and all our collaborators, especially the Gaucher Lab and the Christina Stadelman group, and all the funding sources. I will be happy to answer any questions.
Yes, thank you so much, Shreeya, for a great talk. We are in the Q&A portion of our seminar today. So if anyone has a question, please feel free to drop it in the chat and I can read it for you, or you can raise your hand and ask Shreeya yourself.
Hi, Shreeya.
Hey.
That was an excellent talk. My name is Felipe Scafino. I just have a question about a slide you presented towards the end. I think it's slide 34, where you treated them with interferon gamma. And this was in 5X mice.
Yes.
Could you explain a little bit more about why you don't see an increase in the interferon treated group?
So these are the
relative to control?
microglia, which we sorted out from the 5X FAD. I don't have the image here, but if you look at the paper, we have a panel where we show wild type and 5X FAD microglia, a comparison between these two. These microglia are just so activated to begin with that we couldn't activate them even more. They morphologically look completely different and they are very aggressive. So we just couldn't activate them even more because they were really activated in the first place. Much more than the wild type microglia.
Right. Do you think that if you gave interferon gamma at an earlier time point in 5X that you might see some changes?
So just to clarify, we're not giving anything to the mice. We are giving everything to the cells isolated from the mice.
Okay.
So we isolate the cells and then we treat everything ex vivo. We did not give the mice itself anything.
Okay. Thank you. Excellent talk.
Thank you.
Given that you observe the MSC class 2 positive microglia, do you see any CD4 T cell responses in
I was
mice?
also very interested in this, and I seen for CD4 T cells. In this model, we don't see a lot of CD4 positive T cells. So I don't think this is the best model to look into that. And that's one of the reasons why we never focused on this in this paper, because we don't see them in the first place.
And I would like to hear your thoughts about CD8 T cells. Are they good or bad in AD models?
I might be very biased here. I might be very biased here, but I know that the CD8 T cell field is very divided, and there's a huge literature which says they might be beneficial. There's a huge amount of literature which says they are not. What we are seeing right now with our stuff is that they can induce a lot of myelin damage and oligodendrocyte damage. And we need to further look into what exactly does this damage mean, because this is something which we haven't been able to do so far with this model, is how bad is this damage and what are the consequences of it. Also, I feel like a lot with AD research, it's a timing issue. They might have some sort of protective effect in the beginning, but then this is completely overshadowed by what they start doing towards the end, which is really, really damaging. That's the same with microglia, like they're really protective in the beginning, but then they get really activated and start damaging things.
Yeah, totally agree. Thank you. Today is our last seminar of the year. So if you have any questions, you're welcome to raise your hand and start your video or just put them in the chat. And if you want to learn more about future open science seminars, I put the 2 links in the chat. So one is nominate a speaker, the other is join our email list, and you will get the information about future seminars. So we have a few, maybe 4 seminars lined up in January and February already. So stay tuned. So if no more question, I want to thank Shreeya again. It's a wonderful talk. Thank you so much.
Thank you so much for inviting me.
Yeah, thank you. And then happy holidays, everyone.
Merry Christmas.
Okay, thank you. Bye-bye.
Bye.
Thank you, Shreeya. Bye-bye.
Bye.